So it's my pleasure to kick off the pinnacle speaker of the last session, who's also the beginning speaker of the afternoon. So it's a unique position, only afforded rarely by changes in scheduling. So it's my pleasure to introduce Chantal Stern, who I'll just be very brief so we stay on time. But Chantal, like several of our previous speakers, was one of the bold individuals who jumped into this new enterprise at its very beginning, which, in seeing the successes of the field, it's easy to forget how much courage that takes. But Chantal is a systems neuroscience coming from doing Dick Bassingham's lab, doing monkey work and animal work, and shift to doing higher level cognitive function, and made major contributions, and still has been making major contributions in the field of memory and higher level cognition. I remember several of her early influential studies in looking at working memory, in addition to the work you'll be hearing about today on long-term memory. And I think that it goes without saying that I was influenced by many individuals, but Chantal was one of the few people actually doing work like I had hoped to do in the laboratory when I showed up. And for those of you who might be joining laboratories now or have been in laboratories for a couple of years, you have a very interesting perspective, is that somebody who's been there, even if it's only a week or a year more than you have, is infinitely wise. And so I still look up to Chantal for good reason, but also if I have any questions, she's the kind of person I'd want to turn to because I was imprinted at such an early stage on her wisdom. Chantal's a professor at Boston University, and welcome. Thank you.
Well, thank you very much, and thank you, Bruce, for inviting me to present today. And I'm not really sure whether, I love being referred to as infinitely wise, but when I think back 25 years ago, most people just were telling me I was crazy. They were telling me that coming from a group of sort of a long line of neuroscientists and physiologists, and then sort of deciding to switch and join Bruce Rosen's group as what I remember one sort of saying that I was your first neuroscience postdoc, and once you sort of said, actually, I think you were my first postdoc. But that, it was something that a lot of people sort of said, there's no career path here. There really isn't a career path. There are not people that are already doing this. You can't really look ahead or, look at people that have already gone through along this path and sort of say, what are they doing that you want to be able to do? You really had to look at it and say, this is just really, really exciting. It just seemed like such an exciting thing to be doing. And, we've spoken this morning about Jack's contributions and Ken's contributions, and I really think, Bruce, that one of the major contributions that you've also made to this was in this team building of being able to look and sort of say, well, no, I need a team that includes neuroscientists. And here, we have this group that is primarily MR physicists, and in order to get this to work, we really have to bring in the neuroscience and combine it. And that was what really made it an exciting time back in 1991, which is when I joined the lab. By 1992, it was actually, I was looking through my office, and I still had the original SFN, the Society for Neuroscience abstract books. And I think these are the first fMRI abstracts and talks that went to the Society for Neuroscience. So Ken presented one on the methodology that we heard about this morning, and I presented some work afterwards looking at, well, there's Ken's in terms of describing the technique that we heard about this morning. And mine was really focused on understanding the signal and trying to figure out what what was this strange undershoot that we were seeing. And so while I was asked to speak today, or this morning initially, about cognitive neuroscience, I just think it's important for everyone to recognize that in those early days, the initial studies were not focused on cognitive neuroscience because we really had to just understand what was the signal and what did it mean, and could we use it to somehow track brain function. And as Roger so clearly described this morning, you know, those early studies really did focus on some of the systems that we heard about, so a visual system. We did a number of studies that were done with the motor system where we had people making very simple movements. I had the fortune of working with people like Chris Moore, who was a graduate student at MIT, where we did somatosensory system work. And at that time, I really was also helping people, as Dave Kennedy said, you know, just locate, well, if you only had one slice to look at the brain, where were you going to put it? Like, where was motor cortex? Or where were we going to look if we were going to look in, you know, dorsolateral prefrontal cortex? Or if we were going to do other studies, where should we look? And another thing was just paradigm design, and how were we going to do this in a scanning environment? So when I was trying to put this together, I was looking through my office for old materials, and I found actually the last talk that I gave for fMRI that was actually on a slide projector. And when you think of how much the technology has changed in 25 years, just think that is where we were then. We were putting things together on slides. I had someone come into my office and see this and go, but wait, how did you even make those figures, right? And not only that, the slide projector was part of our experimental toolbox as well. So I was in the scanner room with Roger when we were doing some of those very early MT studies. The MT studies were done with a slide that had a whole bunch of little dots on it. And then Ken would be there, and he'd, you know, someone else would have the stopwatch, and he'd say, go, and he'd pick up the slide projector and move it around, and that was the moving dots. And then stop, and you'd put it down. And then a minute later, go, and you'd move around the moving dots, and you'd put it down. So I know Roger hates that story, but it's how those early studies were done. But you know, one of the things I really wanted to do at the time was try and figure out, well, could we get it to the point where we could use this technique to study higher level cognition? And another thing was really, could we move away from using a surface coil to actually using a head coil and see something that wasn't motor cortex or somatosensory or visual cortex? Could we actually get deeper into the brain? And I had the fortune in the early 90s to actually interact with and then work with Suzanne Corkin, who is a professor at MIT who sadly passed away earlier this year. And I'm sure that Sue would have loved to have been here because she really was one of the people who early on just embraced this technology. She just thought that it was such a cool technique. And you know, as John Gabrielli mentioned earlier, Sue had spent most of her career, he mentioned HM, patient HM, and Sue had spent much of her career studying patient HM, and early on I helped her with some of the studies where we were first doing structural scans on HM and then later trying to do some functional scans with him. I often tell the story that I used to bring my baby daughter to the scanner with me, and she would be in her little baby seat, and HM at one point sort of said, I don't get to see babies very often. And so we picked up Simone and we put her on HM's lap, and he held this little baby, and there were no cell phones, so we have no photographs of that. Being an amnesic subject, he has no memory of that story. Being a baby, she has no memory of that story. But for me, it's a vivid, episodic memory. And that's one of the things that I have been studying for a very long time now. Another thing that really brings home how much things change over the course of 25 years is the fact that this baby now is certified to run a scanner. so that brings home this case that we really can say we've moved on to the next generation of imagers. Okay, so the first study of cognition that I did at the NMR Center was one in which we tried to get hippocampal, and we did manage to get hippocampal activation in healthy young subjects using a very, very simple blocked paradigm. So using our trusty slide projector at the time, we simply showed people a series of complex visual images and asked them to look at each one carefully so that they'd be able to remember it when they got out of the scanner. And this is something that, John described to you earlier in some of the event-related studies, but these early ones were done blocked, and the idea was simply that if you had people look at complex novel information and then you contrasted it with having people simply look at a single image or a couple of familiar images, when they were looking at the novel stimuli, they would be encoding that information, whereas when they were looking at familiar information, there would be no encoding required. And luckily for us, given the fact that we were using a 1.5T system and we were looking for signal changes in the individual subject level, you actually see very robust activity, specifically in medial temporal lobe structures, including the hippocampus, even at a single subject level. And I think that moving forward, when I think about where the field is going, this is really one of the things that we need to be pushing is, again, if we're going to use something like fMRI as a biomarker, can we look for techniques, can we develop the technique in such a way that we can really look at individual differences and that we can look at the individual subject. So, there are a whole host of studies that I have done looking at memory encoding. I'm just going to pull out a couple of them here. I've selected these because I thought they fit better with this afternoon's component on clinical applications for fMRI. So one is the fact that we adapted this picture encoding technique that I just showed you in work that I did with Brad Dickerson, who's speaking a little bit later, where we simply had older individuals who suffered from mild cognitive impairment. We had them do the same sort of picture encoding task and discovered that with MCI patients, you actually see not loss of activity within these medial temporal lobe structures, but actually hyperactivity. So you see this hyperactivity early on, and then later on when people actually have Alzheimer's disease, you see a decrease. So you see hypoactivation in these areas. We've done the same sort of study with different types of dementia. So we've looked at patients with HIV dementia. We've looked at patients with Parkinson's disease. And again, in many of these neurodegenerative disorders, you see this similar sort of hyperactivation followed by a hypoactivation later on. More recently, with one of my graduate students, Yakel Kiraz, who's now at MGH and working in the Martino Center, we set out to do studies in her native country of Colombia, looking at a population of individuals who carry a PS1 mutation for Alzheimer's disease. So these are individuals that develop Alzheimer's disease before the time they turn 50. So they develop it very early on. And Yakel sort of tried to convince me that she should really go to Colombia and run studies in these individuals. And I said, no, this is crazy. You should get your PhD first. You can go as a postdoc. But following in this great tradition, eventually she convinced me, and I said, all right, we just got to do it. So we managed to pull it together. We had lots of help from some of the physicists, including Thomas Brenner when he was at the MGH at the Martino Center, to try and get the pulse sequences to work on a clinical machine in Colombia, and she went down there and tested individuals using this picture sequencing, the picture encoding task in individuals who were in their 20s and 30s. So these are cognitively normal individuals that have nothing wrong with their memory, and yet you already start to see this sort of functional signature, and we've also followed it up with some sort of structural signatures of pathology decades before they actually develop Alzheimer's disease. All right, so at this point I just want to switch directions a little bit, and rather than talking about memory, I want to talk a little bit about some of the more recent work that we've been doing on spatial navigation and goal-directed navigation. And while it may seem like it's a switch from studying memory, it really isn't, in the sense that most of the animal models of hippocampal function have always focused on the hippocampus, and there's a lot of work as well that's been done in entorhinal cortex, looking at what are now known as grid cells. So we know of place cells in the hippocampus, grid cells in entorhinal cortex. And we really wanted to sort of scale some of this work up to look at the human brain. And it also ties in very closely with the memory work that I've been thinking about. So if you think, for instance, of a question of, well, where did you park your car? That can be seen as, if you want to get to your car, a navigational exercise. How do you reach your goal? Where is your car? But there's also components of it, such as how do you tease apart or disambiguate two overlapping memories? So how do you disambiguate where you parked your car today as opposed to where you parked your car yesterday? Or, since it was just Thanksgiving, you can do the same sort of thing, not in a navigational framework, but simply in a situational framework. How do you tease apart or disambiguate your memory for Thanksgiving this year versus Thanksgiving 3 years ago? So in a series of experiments that we've done over, well, I guess this is one from 2010, we've looked at this sort of context-dependent navigation in the scanner. So subjects were taught, in this study, they were taught to navigate through 12 different mazes, and they were taught to navigate these mazes outside of the scanner. And then once they had really learned those mazes, they were put into the scanner and told that they needed to navigate through this space. So in a real-world condition, you might think of something like this. The starting point shown in sort of here would be my office, and you might think about walking down the street and going left to teach my class, or walking down the street and taking a right to go and get a coffee. And if I look at that sort of representation and the trajectory, I need not only, when I'm starting, to plan out my route in space or my route in time, I also have to keep a goal in mind, because when I'm on the street, there's nothing in my visual environment that's going to tell me which way I need to go. So am I thinking back or am I thinking forward? Do I need to do both of these things? And so this is one of the directions that memory researchers have shifted from thinking about memory as simply being able to encode information, being able to retrieve information from the past, but instead to start thinking about this whole memory system as being critically important for what we call prospective memory, or thinking about the future and making decisions and thinking forward towards our goals. And so in this task, participants were taught to either navigate through environments that overlapped with each other. So you can see here, this is a critical hallway that overlaps between these two, the green and the blue maze. And in this case, on the right, you see a non-overlapping trajectory where it's unique. And when we run people in this type of task, we find that when people are initially in this first hallway, where they have to really think ahead to their goal, we see activity within the hippocampus, also within related parahippocampal areas. And when you get to the component where a person has to make a critical decision, so this decision-making process, you again pull in these two areas with additional input from orbital frontal cortex. And so in addition to that, we've followed up on those studies, looking at functional connectivity, and there are different methodologies for doing this. In this case, we were actually using some of the methods developed by Jesse Rissman, but there, you can also use some of the resting state methods that Randy has talked about. And we see connectivity across a network, and I get back to what Randy was talking about, is the importance not of just an individual structure, but the importance of networks of activity for these different types of complex tasks. So connectivity between the hippocampus and the striatal system, connectivity between the hippocampus, the striatum, and the orbital frontal cortex or frontal cortex in this type of task. So, just to finish up, we've also been able to use these sorts of studies to look at learning in the scanner. And this is, I think, again, something when we think forward to where's the field going, can we actually tease apart the variability that we see across different individuals? So in these tasks, we tended to train people initially so that we knew people could do things before they got into the scanner. The problem with putting them in when they can't, when you don't know whether or not they can navigate, and we know navigation is one of the things where we have huge amounts of variability, that's something that, that we've been pushing ahead with. Other things I know I want to wrap up now have to do with using high-resolution imaging, so now we can get to the point where we can look at subregions of the hippocampus and look at differential contributions, different types of parametric correlates for doing these navigation tasks. And if people want to see where the field has really moved in terms of the navigation work, I'd point you towards recent work from one of my former graduate students, Thackeray Brown, who's now out at Stanford with Anthony Wagner, and just published a paper in Science earlier this year on planning and prospective memory. So I'm going to just end there. I think the idea of thinking back is wonderful, but knowing that our brains and the systems are really set up to actually prospectively think forward is something that I'd like to end with. Thank you.